Bio 30s, I'm back again for another lesson. To start, I want to do a little quick experiment with you. So what I need you to do is take both hands and simply just put them together like this, okay? Now, take them apart. Let's do that again, but really pay attention to what thumb goes on top. So we'll do it one more time here. What thumb goes on top? For me, it's always my left thumb. The left thumb feels the most comfortable, okay? Bring them apart. Let's do it one more time. This time though, try putting the opposite thumb on top. So we're gonna do it one more time here. The right thumb, it doesn't feel quite right. Left thumb just feels about best for me, but for you it might be different. So that is actually a genetic trait that you inherit through your chromosomes and your DNA. Now putting your left thumb on top is seen as the dominant version of that trait, and the right thumb on top is the recessive version of that trait. So just a little trick there that you can see if you're either a dominant or a recessive for that trait. So, leading into the lesson today, we're going to talk about dihybrid crosses. We've done some crosses looking at one singular trait. We're now going to look at a cross that includes two traits. All of the rules still apply, but now we're going to do a bigger Punnett square because of it. So, getting back to Mendel and his laws of heredity were pretty simple, right? They said that, or he said that traits are inherited from your parents, okay? They're controlled by genes, and they come in pairs, right? One from mom and one from dad. And then how they interact with one another can lead to the idea of dominance or incomplete dominance or codominance. So in some cases, one gene will mask the other. This leads into the law of dominance, where one allele or one trait will mask the other in the heterozygous genotype. So in the heterozygous genotype, if we have one big R and one little r, in this case for round or wrinkled seeds, if you were heterozygous, you would have round seeds, assuming that it was complete dominance. So for example, we mix one homozygous dominant with one homozygous recessive, we get all of the individuals being hybrids, and these hybrids all have round seeds. Why? Because the round seed allele masks or dominates the wrinkled seed allele. Okay? His second law was the law of segregation. So he came up with the law of segregation, okay? Basically what it says is, during the formation of eggs and sperm, the alleles will separate, okay? So think back to when we talked about meiosis, right? The chromosomes, or the alleles, separate during meiosis. So we have 23 going over here to this sperm, 23 over here going to this sperm, or 23 over here to this secondary oocyte, and 23 over here to this first polar body. So they separate or they segregate. So this individual that is a hybrid could either donate a big R allele or a little r allele. We don't know which one they gave. And if you are same thing over here, they could donate a big R and they could donate a little r. You don't know which one they gave. And then when they recombine, this is how we get our variety of traits. So maybe two big R's combined. Maybe a big R and a little r, maybe a big R and a little r, or maybe a little r and a little r. And that gets me a ratio. And if you remember from the previous lesson, what would that ratio be? A 1 to 2 to 1 ratio. So, law of segregation, simply during meiosis, the chromosomes will segregate or separate, which means you're getting one allele from each parent. So how do we apply that? Well, we apply it in the Punnett square, right? So we have, let's say, for example, tall or short. Female parent being a heterozygous individual could donate the tall allele or the short allele. Once again, we're not sure which one they donate until actual fertilization occurs. Male parent, they can donate the tall allele or the short allele, and now we have 75% chance that the individual is tall, 25% chance that the individual is short. Of course, our genotype ratio being 1 to 2 to 1, our phenotype ratio being 3 to 1. Okay, and the final law I want to talk about here is the law of independent assortment. This is very important. Basically, the alleles for different traits assort themselves independently of one another, and this is what leads into our dihybrid cross discussion today. What does this mean? It means your eye color has no connection to your hair color, right? They assort themselves independently. So you could be dominant for eye color, but recessive for hair color. You could be dominant for your earlobes, or you could be recessive for the length of your fingernails. All of these traits are independent of one another. Now, that's not saying all traits are independent. Some traits actually are found on the same chromosome and they're linked together. So for example, you might see someone with, say, red hair, freckles, and fair skin. Those traits tend to be linked together on the same chromosome. But usually, when we're looking at traits, especially on different chromosomes, they are independent of one another. So whatever happens on one trait does not impact the other trait, and that's the law of independent assortment, right? So when we look at how chromosomes assort themselves independently, we can have our cells going through meiosis 1 and then of course meiosis 2. And when we look at this, right, this cell right here has a big G and a big Y. This one has a big G and a big Y, but this one is a little g little y, little g little y, and then so on and so forth. So what you get for the G trait does not impact what you get for the Y trait. So they assort themselves independent of one another. These two traits are completely different. They would have no linkage whatsoever. So this of course leads into dihybrid crosses where we're looking at two traits assorting themselves independently and they're located on different locations or loci on the chromosomes. So same laws apply as a monohybrid cross for this dihybrid cross. So let's get into it. What are we going to do here? Well, let's say we're going to cross yellow seeds that are round with green seeds that are wrinkled. All right, yellow being the dominant trait and round being the dominant trait. So when we take a look at this here, we have a homozygous individual for both traits. Homozygous individual for both traits, double dominant, double recessive. Well, it's a pretty easy cross because we know they're all going to be hybrid for color and they're all going to be hybrid for shape. So we end up having all of our individuals being yellow and round. So that's a pretty simple cross, right? We know that when we mix two individuals, one homozygous dominant, one homozygous recessive, they end up having all hybrid F1 individuals. So let's do an F1 cross. Let's see what happens when we do an F1 cross, okay? When we do an F1 cross, this is where things get a little complicated. So I'm going to break this down into steps for you. Step 1, you want to write out your allele symbols. Now, I don't think you need to do step 1, but if you're having trouble organizing these problems, I recommend you do step 1. So what I've done here is I've wrote out my symbols or a little legend for myself. So I know that big R is for round, little r is wrinkled, big Y is yellow, little y is green, and remember, these traits have nothing to do with each other. So oftentimes I'll see people write genotypes out like this. This is wrong, okay? Those traits don't interact with one another. To write a proper genotype, you must do this. You have to keep the two alleles together, so that's correct. So, step 1, write out a legend if you're having difficulties organizing your questions. Step 2, you want to write out the genotypes of your parents. So that's what I've done here already. So the genotypes of my parents, once again, we're doing an F1 cross. So there's the genotype of parent 1 and the genotype of parent 2. Hybrid for round, hybrid for yellow, hybrid for round, hybrid for yellow. Step 3, write out your allele combinations for the gametes. These are not genotypes. These are gametes, meaning they are essentially half a genotype. So let's think about this, okay? This individual right here could donate a big R. They could also donate a big Y. So that's one possible combination that they could have. They could donate a big R and a big Y. They could also donate a big R and a little y. So that's a second possible combination we could have. They could donate a little r and a big Y. That's a third possible combination. And they could also donate a little r and a little y. So an individual that is heterozygous for both traits would have 4 possible gamete combinations. Big R, big Y, big R, little y, little r, big Y, little r, little y. And this individual would have the same gamete combinations. So we need to make sure that we're writing out all of the possibilities, right? All of the possibilities. All right, let's set up our Punnett square. It's a little larger than normal. Why? Because we need to put all of our possibilities on the top and the side. That means we have 1, 2, 3, 4 possibilities here, 1, 2, 3, 4 possibilities here. Instead of having a 2 by 2 Punnett square, we now have a 4 by 4 Punnett square, giving us 16 boxes in total. Now, we would fill it in just like we normally would. So for example, we match these two up, big R, big Y, big Y. So this individual would be round seeds, yellow color. Over here, big R, oops. Big R, and big Y, little y. This too would be round seeds, yellow color. So I'm not going to do all of it for you because I've already done it here. We fill in all the genotypes and then afterwards we can get our phenotype ratio. Genotype ratio is going to be a little difficult here, so I'm just focusing on the phenotype ratio. If we look at the phenotype ratio, and I'll do this in different colors here, you'll notice that for round and yellow, so round and yellow, we should have 9 individuals. Let's count up 9 individuals that are round and yellow, okay? This individual right here, round and yellow. Round and yellow, round and yellow, and round and yellow. There's 4, okay? This one right here, round and yellow, yep. This one, nope, 2 little y's, so that'd be green, so that wouldn't count. There's another one. This one would also be green, so that doesn't count. This one's good. This one's good. No good, no good. This one's good. That one's no good. That one's no good, and that one's no good. So now we have 1, 2, 3, 4, 5, 6, 7, 8, 9 individuals that are dominant for both traits, round and yellow. Okay, what about round and green? Let's take a look at round and green here. There should be 3 individuals that are round and green. So we need at least 1 big R and 2 little y's. There's 1, there's 2, and there's 3. There are 3 round and green individuals. Okay, let's move on to our wrinkled and yellow. So wrinkled and yellow. So this would be 2 little R's and 1 big Y. Right there, and right there. And finally, we only have, out of 16, just erase that there. So out of 16, we only have 1 individual right here that is wrinkled and green, so double recessive. So 1 out of 16 chance, so very unlikely, but still possible. So we look at it all together here, 9 individuals having the double dominant phenotype, so round and yellow, 3 individuals being green and round, 3 individuals being yellow and wrinkled, and 1 individual down there being the green and wrinkled. So once again, all the same rules apply. We just have to make sure that we're writing out all of our genotype combinations leading to our allele combinations. We put them into our Punnett square, we match up our genotypes, and then from there we can get a phenotype ratio. Now remember how I said that the 1 to 2 to 1 ratio was one of the ones you want to remember? This is another ratio that you want to remember as well. Very common ratio when we're doing dihybrid crosses, a 9 to 3 to 3 to 1 ratio. Once again, this is a phenotype ratio, not a genotype ratio. So that's all I have for you today. Next lesson, we'll be getting into traits on the X chromosome. So we're going to look at X-linked traits, and we'll talk a little bit about how males and females differ because of it. So thanks again for watching, and we'll chat soon.